I'm very happy to be able to share some thoughts with you on this 28th tRNA conference event. I was at the 1972 tRNA conference in Princeton, New Jersey, which was my first. And I believe there was at least one before that, which my guess is Dieter Sohl attended. So he and I are probably the only ones amongst you who were at the 1972 one at Princeton. And he was the only one of all of us at the earlier one. I'm going to talk to you about entering a brave new world, a brave new tRNA world, and one that I'm quite excited about. tRNAs, we worked on them from the earliest days after their discovery. Sequences, structures, recognition, on and on it went through the 70s and 80s. And at some point, many people in the field said, we know everything there is to know about tRNAs, so why keep working on them? And they left the field. And I myself was amongst those who was questioning, what more can we do? And to find the answer to that, as I searched in my mind, where really should we go? Where should I go? I often have reflected back on a movie that came out 40 or 50 years ago called Back to the Future. The lifelong question of what is the road ahead in our professional life and in our personal life. What is that road? And really, the statement by the character Doc Emmett Brown, where we are going, we don't need roads, always stuck in my mind. Because it actually is life. When we go back to the future, we're going back to our roots, ourselves. When we first started out, full of anxieties, and we didn't have any roads, and we weren't sure we even needed roads. We were trying different things. But at the same time, we had this sense, we've got to find a path ahead. But we need to have this wide open sense of freedom. We don't want to be pushed on a particular path. We want the freedom to feel we don't need roads. And we have to go through this process throughout life. There are no roads on a hockey rink. When I was very young, I followed hockey. But a neighbor of mine still follows hockey and told me about a player who really epitomized what I was looking for and what I have looked for over the years and still do. The player was Wayne Gretzky, who was widely voted by all the newscasters and sports writers as the best hockey player of all time. He did not come in as the heaviest, biggest, smartest, but he did have one characteristic that set him apart, that made him the greatest. And it's a characteristic applicable not just to sports, a characteristic held by Elon Musk, Steve Jobs, Jeff Bezos, and other great entrepreneurs of our time. He did not skate for the puck. He did not skate for the familiar. He did not skate for what's obvious. You might call it the comfort zone, but he consistently anticipated where the puck was going to be, and that's where he went, and that's what made him great. When I look back at myself, I think of myself as kind of a failure in that respect. because I did not anticipate, I was very skeptical of efforts to jump into tRNAs as therapeutics for new medicines. All tRNA, when they made their announcement, I know Flagship Pioneering. I know the chairman and CEO, Nubar Afif, and a Armenian refugee who grew up in Lebanon and migrated to the United States. He developed Moderna with some of my colleagues who I knew well. I was very skeptical of Moderna, injecting messenger RNAs into people. But he believes that tRNAs are now the next big impossible mountain. It's where the puck is going to be to treat thousands of diseases. And Peter Deaton has started an enterprise and gotten it nicely financed with his former student, Nick Davis, and some folks at Harvard. I believe they're greatly interested in modifications. And Tevar Biosynthesis Sciences, started by Harvey Lotus and a couple of fathers, experienced businessmen who each have a daughter with the same very rare form of chronic, nearly acute epilepsy, for which there is no medicine. Devastating. And that is caused by a nonsense mutation in a particular messenger RNA that affects the behavior, functionality of the brain. And they believe they can deliver suppressor tRNAs into these people and rescue them. Remarkable and doable, I learned. Here's a paper that came out in Nature just recently using AAV suppressor tRNA to overcome a nonsense mutation in mice. They use AAV, but I had learned at Alnylam through the extensive investment we made in delivery that lipid nanoparticles work very well as well, especially for our targets at Alnylam, for RNAi. So conquering the delivery problem is what Nubar saw, which they did at Moderna, and which will be done now by many groups around the world to use tRNAs, including suppressor tRNAs, as therapeutics. Very exciting. And this, of course, has been picked up by the news and the trade magazines. And recently, not highlighted here, a very nice extensive article in Nature magazine itself. This is where the puck is going for the brave new world of tRNA. Nature Cell Biology. This is a cover based on the work of Qi Chen. Expanding universe of small RNAs. Where do those small RNAs come from? Some of them come from tRNAs. Their fragmentation to generate many regulatory molecules. We call them tsRNAs. Some of these do look like and appear to be microRNAs. Some of them can be non-coding RNAs that regulate genes. Roughly 70%, maybe more, of the genome is regulated not directly by proteins, but by RNAs, like microRNAs. Here is an example, a paper you should all read for its thoroughness and depth of analysis. Oxidation of a miR1 microRNA. Converting a G to an 8-oxo G facilitates a GA base pair. That GA base pair now enables this oxidized miR1 RNA to misseed at a site that changes the architecture of the heart. This is a epigenetic phenomenon caused by oxidative stress, for example. And humans can be diagnosed for it. And it's a rather large number of people with hypertrophic cardiomyopathy that have that myopathy, by all appearances, due to oxidation of miR1 RNA. I'm not saying this came from a tRNA, but it is an example of the kind of things that can happen with tRNA fragments. Now, we know that one of the most exciting developments in our time in the last 5 years has been the deep learning progress made by Google DeepMind as well as many others. But we learned a bit about this through our efforts at the Laboratory of Hong Kong in collaboration, and ATAR, in collaboration with Nancy Chung at Columbia. This is a case where a mutation in a human inherited by offspring in the right context develops a large number of comorbidities, with death ensuing generally within the first 12 years of life. Work of Juwen Xu, Carrie Low, Leslie Nangle, Nancy Chung teased out some of the effects of this mutation using patient samples. And here is a result from RNA-seq analysis that we filtered out all the noise. And these were some of the things that came up that were well above the noise. Reproducible and statistically significant. And you can see that it's a vast network of interactions between many different signaling pathways. And yet this is only one layer of what's happening. And it is all about the cell making choices to try and find the path to homeostasis. Very interesting and very overwhelming in some ways. So I sat back and reflected and asked myself, can I imagine anything that is not necessarily biology or protein structure like the AlphaFold analysis, but rather just in real life that exemplifies what can be done if you can make the right choices using the power of machine learning and deep learning, which the cell undoubtedly went through over many tens of millions of years to get to this very complex state where you find the path to homeostasis. And for that, I was struck by an article I came across in Bloomberg Business Week, which came out about 2 to 3 years ago and was very well vetted and researched. But it was an article documenting how an individual picked horses so successfully that he made almost $1 billion betting on horses. And starting out with a 15% handicap against him at the house. That is to say, the house taking 15% no matter what. Very difficult. But this is Bill William Venter. But he worked and worked to study track conditions, jockeys, temperatures, moisture content, and developed through trial and error and machine learning algorithms that made him enormously successful at betting on horses. So I was actually very encouraged by this because he found a way in what would seem to be an impossibility to choose the right horse. I couldn't believe it. But I thought to myself, well, this is on a much grander scale, what the organism is doing to survive. And for that, I turned back to try and, in my mind, picture how cellular intelligence evolved through eons of training and deep learning, particularly thinking of tRNAs and all the millions and millions of years of training that tRNA went through to develop all these multifunctionalities. So here you see perhaps something you're familiar with. You have inputs on the left and outputs on the right. You have an initial outer layer, and then you have all these hidden layers. Hidden layer 1, 2, 3, then an output. So on the left, the input could be a mutation that causes Charcot-Marie tooth disease. On the right is the output, CMT. On the left could be high consumption of sugar. On the right could be diabetes as the output. Going through all these hidden layers that we don't really understand. On the left could be exercise as the input. On the right, the output, low blood pressure. So you have this outer layer, which we can usually all see. We see the receptor for sugars, for example, but the rest of it we don't see. And there are many more layers and layers stacked on layers. This is what cellular intelligence evolved through over the eons to get trained in deep learning. I looked in the literature for an example of how this might be applied now in biology. And this paper was called to my attention about how studies of a deep neural network for prostate cancer, deep learning, led to many more insights into the etiology of prostate cancer and the enormous complexity, as only one cancer out of many. And this example pertains, of course, in a different way, different layers, to breast, the colon, the lung, and so on. This group at Harvard uncovered 6 layers in prostate cancer etiology. There are more, but here you go. On the left is the inputs. On the right, at the far right, the outcome, prostate cancer. On the left, inputs, amplification of a gene, mutation of a gene or genes, and deletions. And then you see H1, H2, H3, and so on, all these hidden layers, which they teased out in a beautiful paper, beautifully illustrated, to eventually see the pathways to prostate cancer. And now take this information to take a patient at the front end at diagnosis, figure out what the input is, and then understand better the prognosis and the particular type of therapy and approach that should be used to move through these layers and divert some of the pathology and improve overall survival. A fascinating piece of work. Let's go on and talk about the Y base. This is probably, in my mind, the most famous modified base found in phenylalanine tRNAs, Y-butyrosine. I say famous because when I started out, it was the big modified base, not only because of its size, but because it was fluorescent. And this enabled people who were very interested in doing physical measurements, fluorescence polarization, fluorescence relaxation times, and so on, to study a tRNA in solution with a marker on the molecule that provided information. I remember how Nakanishi at Columbia, where I spoke to at the time, was very much involved in this, as were many others, but really enthralled by what you could do. That was 50 years ago. And that was it. That's the Y base. No one thought about really what it might be for. It was just there. But Peter Deaton pointed this out to me, how a modified base, particularly Y-butysine, connects to patients afflicted with colorectal cancer. Now, in this example of a waterfall plot of overall survival versus seriousness diagnosis, you'll see that it starts with methylated CpG islands. Well, you know, if you heavily methylate epigenetically, CpG islands, the genes associated with expression at those islands will be shut down. So if you methylate CpG islands associated with one of the 7 enzymes involved in Y-butysine synthesis, you will shut down the production of the Y base, and you'll have tRNA feeds that lack Y. And you can see the ones that are left alone, not heavily methylated, epigenetically unmethylated, so the Y base is retained, have a much longer survival, much better outcome. And this really was teased out in this paper that Peter called to my attention, and which we discussed in our journal club at Scripps, by a group listed here from a number of institutions around the world. But what they found is that this lack of Y leads to frame shift mutations, creating a premature stop codon in particular in a tumor suppressor gene known as roundabout receptor, ROBO1. And that in turn generates a messenger RNA decay via the nonsense-mediated messenger RNA decay pathway. Exciting. Many layers had to be gone through to get this result, but also incredibly insightful of the subtleties and the deep layers that are associated with tRNA biology in this brave new world. So let's find the hidden layers of sarcoidosis. This is a project that was initiated in Hong Kong with our group there, financed in part by the Hong Kong government. Leslie Nangle, Terry Lo, Juan Shu, Shan Liang. We set out to discover splice variants of tRNA synthetase, and we found over 200 of them. And that eventually led to our interest in sarcoidosis. This image is a lung in the terminal fatal stages of interstitial lung disease, filled with sarcoid tumors, as you can see, scars, scaling, and now dysfunctional. What we discovered through many cell-based assays, that this splice variant, an N-terminal fragment of histidine tRNA synthetase, was a potent therapeutic. How do we find this? Simply by screening many cell-based assays, different ones. I believe there were 30 different ones. At ATAR, Elizabeth Gardner did a lot of this work to identify signals coming out from the splice variants that were associated with particular biological pathways. Amazingly specific in this large matrix of over 100,000 data points that we generated. But this WEP domain turns out to be a potent therapeutic. Here's work that was done by Leslie Nangle and Ryan Adams and their group at ATAR. This is work in animal models. On the left, you can see a healthy lung. Inflammation and fibrosis are absence, top and bottom left. In the middle, the injured lung, you can see enormous inflammation and all kinds of fibrosis. When you apply this WEP domain as an FC fusion, we call it ATAR 1923 internally, you can see remarkable resolution. When you carry this forward into humans, which is where we go, here is one of many images I could show, data from a phase 1b/2a trial, that on the left, without needing to read through all the fine print, you'll see a number of things that were tested. And on the right, the different colors represent the result in humans. It's not important that you see all these details. You see 4 boxes across the top here. These are different doses with a placebo on the left, and then gradually increasing doses. And you'll see it gets greener and greener as we go to the far right. Remarkable effectiveness. This now takes us into phase 3. And we're now beginning the process, slowly but surely, to recruit patients for that purpose. But this is also, in my opinion, as a partner of tRNAs, the synthetases, a brave new tRNA world involving now splice variants, which maybe are analogous to the sRNAs, the fragments of tRNAs. So, tRNAs. Why keep working on them? And really, this is one of my favorite quotes. It's what you learn after you know it all that counts. John Wooden, the former coach at UCLA, who won 10 NCAA titles in 25 years. The next 25 years, they won 1, I believe. Coach means a lot. Lab head means a lot. But he had players coming out of high school to him at the UCLA Bruins, and suddenly thought they knew it all. He knew better, and he taught them. You're only in the first inning of learning. After you know it all, what you learn is what really, really counts. Thank you so much for sharing this time with me. I was so happy to be able to do this and have this opportunity. And I very much look forward to learning about all the wonderful things you did at the meeting, the talks, the discussions, and so on. And wish all of you the very best as you enter this brave new tRNA world.